question and answer session.
Right, right. Okay, so I thought what I will do is well, first tell you a bit about myself, my past, to how I end up doing what I do, and then maybe generally think together with you about physics, biology, what does it mean to be at the interface, right? And then now I have, no, I of course I have my research stuff, and if you're interested, I can tell you a bit about research, maybe give some illustration, but that's not really the focus. And the focus really to, I do not know what you look for, especially. So as we discuss, you ask questions, and I will have a better idea of what would you would be more interested in, right? And then just take things from there. Okay. So I came to UCSD 25 years ago. Long time ago. Most of you were not, all of you, none of you were born yet. And yeah, and I came to the physics department actually as a plasma physicist. Just so happened to be that's an area they were hiring. Okay, even though I did not do any plasma, but generally I was working in the area of complex systems. Okay, that's how I got started. And so then a few. So I was trained as a theoretical physicist. So my Ph.D. is actually in statistical mechanics. Okay, so we're looking at the phase transitions, non-equilibrium phenomena, sort of a general how simple models, simple system can generate complex behaviors, right? And then I started. So as I came to, one of the reasons I came to UCSD was I know that biology is big here. But even back then, 25 years ago, okay, so at that time, most physics departments do not accept biophysics as a branch of physics. Okay, UCSD was one of the few exceptions. And even though I had not done any biophysics during my PhD, I thought, but that's the direction I wanted to go into. I didn't know what quite it would be yet, but I thought it would be very interesting, and I moved into it. I came here with having in mind to move to biophysics. Okay, and so then my first 5 years, I was mainly just doing sort of theory, statistical mechanics applied to biology, and then somewhere along the line, I picked up a lab. So I was a theorist, but picked up a experimental microbiology lab. Okay, and so we had the lab started 2003, so it's been 17 years now already. Okay, so my group now, we do a combination of experiments and theory. Okay, so the experiments are all table type experiments that even theorists can do. Okay, so many of the people that come to my lab started as theorists. They got interested in sort of question. They want to see what the reality is like, and they do some simple experiments. We figured if the experiments that we use very old technology, 1950 technology, and to do very simple measurements. Okay, but then we ask. So what's I guess what's different about us? So we ask questions that biologists would not normally ask, and then by having some data, then allow us to sort of pursue and look at biology in a different direction. Okay, and so we have so currently we have about a dozen people in my lab. We'll say about 3-4 people only do biology experiments. They're from biology training, biology student, biology postdoc. But there are already people that come to my lab, even for the biologists, they're kind of self-selected to be more quantitative minded, right? And 3-4 students would, 2-3 students right now, they're theorists. Okay, they don't work in experiment, but they interact well with the experiment. They understand how data is taken, what the weaknesses are, so forth. And then about the rest, about half of the people, they do both. Okay, and that's the majority. Okay, and the bulk will be physics PhD students and physics postdoc. Okay. So that's kind of my where our labs are now. And so it's a combination of theory and experiment from a methodological standpoint. Maybe, so to talk about biophysics, I think it'll be fun to first think about together what is physics, right? Well, so tell me what you think. What is physics? It's the study of blah blah blah. All right. What do you think? What did anybody ask you what is physics? What do you know? Anybody?
I think it's a study of how the world works, but with physical objects.
Okay, so it's a study of how the world works. That's probably what I would have said if I was asked that. Okay, but then there are many studies of how world works, right? The world, let's say we talk about inanimate world, certainly, right? So before getting into biology. So, very early in history, Aristotle was thinking about how world works. Now he has a theory of of different elements, fire, earth, and so forth. You call that physics? Probably not. Right? It's just thinking about how the world works. I mean, theorists are thinking about how the world works, right? And then if you go further, you have, before Copernicus, there's this way of thinking about how stellar motion occurs. So there's a person called, I don't know how to say, Ptolemy. No, if you guys may have seen a very elaborate way of how sort of stars, planets move around in the sky. Right? So this guy tabulated how things move around, right? And has a very elaborate picture of things. But he's thinking about Earth is at the center, right? And all the stars and everything goes around. Okay, so this guy's thinking about how the world works, right? We probably do not call this person a physicist. Maybe the first person close to it may be Copernicus, right? Who's the first one to suggest maybe sun is the center of the solar system. Would you call Copernicus a physicist? How many people call Copernicus a physicist? Nobody? Okay, I'm not sure whether I call him a physicist. Okay, what I'm getting to is, so yes, the different ways to think about, to study how the world works, but not every way is physics. Let's think about another way, chemists. I mean, they study how the world works. I mean, chemists are chemists, not physicists, right? So yes, the physicists, we study how the world works, but there's more to it than that, right? So who would you think of as the first physicist? I mean, think about the famous people, Newton, Einstein, whatever. Okay, no, they're obviously physicists, right? Who would you think of as the first person you can think of early in history who's a physicist? Before Newton, if you think back to physics 2A, your high school physics. Newton's most well-known things are Newton's law of gravitation, how planetary motion goes around Earth, right? But before Newton, there's another name. Remember?
Kepler?
Kepler. Johannes Kepler. Kepler's law, right? R cubed goes to T squared. That's the planetary motions in the form of an ellipse. Okay. And actually, slightly before Kepler, there's another person, a great physicist. So this we're talking about around beginning of 1600, late 1500. That was really the beginning, the renaissance of many things, including the study of science, right? So
Galileo was
Galileo, right? He is really credited as the really the founding father of modern science. Like, and of course, we physicists rightly pride Galileo as a physicist. Certainly, I don't think anybody would not call Galileo a physicist. Right? So what's the difference between Galileo, Kepler, and Copernicus? They're all studying. Galileo made the first telescope and looking at planetary motion. And what Kepler did was that he took lots of data curated by Tycho Brahe, whom he studied with, right? And by analyzing, looking closely at these the data on planetary motion, he came up with his law. Right. So these at that time, I mean, there's one of the few things you can be curious about looking out in the night sky, and right. So different people look at different way, and certain way of looking at it we call physics, right? So what's special about it? Right.
I think
what's
what's
special?
different with each of them is the tools that they had and the knowledge they already had and what they were building on.
Right. So yes, that's the generally in science you need instrumentation to make observation, right? And then you take the data and you do something with it. So even before Copernicus, the guy I talked about Ptolemy, I don't know how to pronounce this. Now he was also observing, right? Here's the data and he's doing something. But the big difference between Kepler, Galileo, and the earlier ones, right, is they actually associate physical objects, laws of nature, with mathematics. Okay, Copernicus is saying in word, yeah, things going around it, so you can push that to mathematics, but it's a concept, right? And before that, Aristotle is studying action. But Galileo was the first person, like who actually believed it existed, that laws of nature, right, can be described by mathematics. Like that's a huge progress. Why should mathematics have anything to do with what you see out there? No, really think about it. That's amazing, right? And Galileo was also the first one credited with the scientific method that started with him. Many things he understood the difference between. He knew that physical laws of nature have something to do with mathematics, but that he also could distinguish between theory and mathematics and the theory and experiment. And he was doing all of these things. Right. Okay, so then, and of course, then after that, and then it gets easier, we have the Newton, right, that developed special mathematics. Before people were mapping the world to something they already know in mathematics, right? Kepler, T squared R cubed, something already, okay, ellipse people already knew, right? But Newton actually developed mathematics so that it could be used to study the physical behavior. Right? So that's taking another step. And okay, so then physics gets going. Then let's see. So that's 1600, right? And then 1700. 1700 is kind of boring. Mostly it's development mathematics. Laplace, Lagrange, these people. Yeah, lots of progress in mathematics. Okay. Now 1800. Anybody recall what happens in 1800? Think about actually in terms of the society. Right? You guys all took your first year of the humanity and so forth, right? So in 1800, what's the big thing?
Are you talking about the Industrial Revolution?
Exactly. 1800 Industrial Revolution, right? So you think about Industrial Revolution, you think about chimneys with heavy smoke pumping out. You think about trains, you think about steamboats, treadmill, and all that stuff, right? So what's going on there? What does physics have to do with it? Industrial Revolution, train, steam engine, right? Heat engine. Okay, so 1800, right? So what we see in the Industrial Revolution is the output of a fundamental understanding of nature, and that fundamental studies is in the form of a lot of it is say gas laws, right? Entropy, thermodynamics. That's the physics that's happening. Okay, and if there were anything, it's actually it wasn't really physics, it was just heat engine. Engineers tried to develop better heat engine, but it became when you really dive into it, it became a branch of fundamental physics. Right, the notion of entropy is as fundamental, one of the most fundamental notions in physics. Right, so we have this Industrial Revolution happening together. It's hard to see how one can do without the other. right? But then they will go hand in hand, advancing technology, advancing basic science, right? And if you were to associate some names to the development of physics in 1800s, who would you come up with? Say thermodynamics. Who? Anybody? Any names? Entropy? Any names?
Maxwell.
Okay, Maxwell is the late 1800s. He is certainly a giant of a giant physicist of the 19th century. Right, but Maxwell is more on the other side, looking at electricity and magnetism. So that's a parallel movement that's developing in the 1800s. People like Amperes, Watts, Volt, they're all in the 1800s, right? So, and that eventually led to Maxwell's unification of these ideas. But then the other side, there's so Maxwell would be one of the two giants I would associate with the 1800s.
Boltzmann.
Huh?
Boltzmann.
Boltzmann. Yeah. Right. So Boltzmann is the one that connected what's happening at the molecular scale to the gas laws and everything at the highest scale. Okay, so now we move to 1800, go to 1900. That become much easier. What's a big thing in 1900? Early 1900, revolution in science.
Relativity.
Okay, relativity is one. Einstein wrote 3 papers. One is relativity, and what's the other 2? 1905, 3 papers by Einstein. Right, that changed the world.
Quantum.
Relativity. Huh?
Quantum physics.
Quantum, right? So that's the photoelectric effect, right? He got a Nobel Prize actually not for relativity, not for anything else, it's for photoelectric effect. Something that requires one line of it's not even algebra, right? It's just, so this shows you can be a theorist without the need to do really fancy mathematics. It's the idea. Einstein is just so creative with ideas. Right? Ideas and mathematics are 2 different things. Okay. Yes. So relativity, quantum mechanics. Now, really, quantum mechanics are really what defined the 20th century. Right? With quantum mechanics eventually giving rise to solid-state physics, electronics, and all the things we enjoy today. You guys enjoy, just take for granted, with Wi-Fi and iPhone and everything that came from studying quantum mechanics. Right. Okay, but the fundamental idea is the quantum mechanics, right? And the quantum mechanics studied how? Do you guys remember? Did they tell you about history of how things get started? Do you remember a course in quantum mechanics? What's the first thing they teach you? You don't randomly just like proposing wave function or things like weird things like that, right? How did they get to it?
Would it be the ultraviolet catastrophe?
Ultraviolet catastrophe. This weird phenomena that nobody links to anything, has nothing to do with Wi-Fi, right? Ultraviolet catastrophe is about black body radiation. You have some heated body, and the light emitted from the heated body is different from the classical excitation. There's some difference, right? And by pursuing that, you get to something. And another aspect is the line spectrum that's emitted by metal. So if you take a piece of you guys probably, I don't know if you've done this experiment. You take a piece of, say, sodium or something, right? You put it under a Bunsen burner, right? And it excretes some light. It emits some light, okay? And if you can see that, you can resolve it by using a spectrum, right? And you see discrete line spectrum. You take a different element, it gives you another set of line spectrum. So back then, so there's a proliferation of this line spectrum, and people had no idea what to deal with it, right? So these material people that were characterizing this line spectrum, back then they had a telephone book thick kind of a catalog about line spectrum. Okay, so these are just unexplained, very complicated seeming things, just detail of the materials, right? But ultimately, that led to Bohr's model of atom and understanding of this thing, right? So ultimately, you understand that entire telephone book worth of a line spectrum can be collapsed to a simple formula, right? You put it in your pocket, and you just know it, and that's it, right? And that what's that? It's basic understanding that line spectrum is a difference of energy level. And it's a simple way to describe energy, right? So you see how sort of things progress, and how physics claim areas that has nothing to do initially with physics. Okay, so physics expanding, right? Reaching its hand to the very, very distant area. And then later in the area I'm going at, polymer physics. Polymer becomes a discipline of physics. Okay, and many other things. So we study non-equilibrium system, chaos, many sort of physics, domain physics keep on expanding. Yes, we don't claim all areas. We can say, oh, that anything you study physical world is physics, right? Well, we said it's not. So chemistry, physicists do not claim chemistry as physics. Why is that? Right. So I'm sorry for this long thing because I'm getting to biology, right? So, but we need to have an understanding about what is this physics, what is this not physics, right? So why we leave chemistry out? We don't want to claim chemistry as part of physics. No, there are areas of chemistry that's very close to physics, physical chemistry, spectroscopy. Right? But generally, chemistry is chemistry. Well, the topic of and you can say, well, okay, so I said I gave a criteria of physics is a quantitative description of the world, but chemistry is also quantitative. Right? You have an IR spectrum, this and that, and you can put numbers on many things, reaction rates, and all that. Right? So then, what is why is it not chemistry? Material science, it's not chemistry. That's not physics, right? Studying properties of all kinds of material. But certain part of material science is chemistry, is physics, superconductivity. Right? So certain materials, when put to low temperature, below zero temperature, it already finite temperature, went through transition, it becomes conducting without any resistance. Right? That physicists came under physics, condensed matter physics, with many study of this type of thing. But certain things, that's just materials. Okay, so basically, there are phenomena where as an example that leads to sort of a conceptually whole new class of phenomena, a whole example of a whole world of a sort of new relations. Right, the new possibilities. And there are phenomena, materials, where it's just kind of a complicated version, sort of just details, right? And so physics somehow kind of shove away the latter part and, oh, they're just details. But these details, of course, can be very important in an engineering sense. And a lot of physics trained people go to study them, right? You see that distinction. Okay, so basically, so physics is the two type of so these are called applied physics. So people use method developing physics to study those phenomena, to study this material, right? And it could be very important for medical, for this and for technology, right? So could be very important. But they're not giving us kind of a new type of phenomena, conceptually new, different types of phenomena, right? And then we call that applied physics, right? Whereas the other type of stuff, by studying certain material, by studying certain phenomena, it leads to new conceptual breakthrough of the possibilities that you see not only in this system. Once you understand it, you recognize it, you're seeing across systems. Okay, so there's two type of the sort of ways physicists engage in studying the physics. Okay, so now enough about physics, and let's talk about biology. What is biology? Anybody? Those of you studying biology, what do you say is biology?
The study of life.
Study of life. Study of a living system. Right. The difference between physics and biology. Well, physics, at least so far, is confined mostly to study inanimate, the inanimate world, right? And biology is about the study of the living. Well, how do biologists study biology? How do biologists study living systems? So. Actually, you have some experience now studying immunology. Can you describe how biologists study it?
Well, I know at least for what I'm doing, and we're looking at the stuff that I do, the lab is looking at malaria and immune response in patients with malaria.
Right.
So in that case, they're doing things like taking blood samples.
Right.
And then looking at what type of things are found in the blood, like white blood cell count, red blood cell count.
Right. right?
Stuff like that. So we're
Right.
observing things, taking samples of stuff and observing and collecting data from what we see.
Then what do you do with that? Once you see the stuff that's in the blood sample, what do you do with it?
Well, we try to understand what it means. Well, at least with this, like everyone with something that's as complicated as the immune response,
Right.
We're trying to understand how does an infection, for example, affect the body? How does it affect, like what type of cells are being brought in to help protect the body? What cells are being destroyed? We're trying to understand
Right.
like what systems, in this case, in the body. There can be other biological systems that exist.
Right. So very good. The many studies of biological system is you go in, you characterize what are the components, and you do something how it changes when you compare something to control. You treat it in a certain way, see how things change, right? And so often in these kind of studies, how do you represent your results? Do you represent, you have some finding, right? Eventually, let's say you write up a report, you present it. In what format do you present the result? Do you present the result as a equation? No. Do you present?
Okay, well, I was gonna answer your question as no, but then it depends, because what I do also is we use what I'm starting to do is to use differential equations to map the immune response, so that if we can better understand how an infection affects the body, then we can better engineer like treatments. So
Right.
we do use math to explain some things, but that's just for what I do. You can explain things in
That's
many
why
different ways.
you're recruited to the project, because you're physicist, right? So what you described, the way biology interacts with math when it does, in your case, is you're using a mathematical model try to explain some feature. But what I'm asking is just the result, how you represent it. Right. So usually, I tell my experience, if you look at biology papers, I do not know how many of you are sort of deep into biology. So it's very rare you have XY plots. So physics is always like you have some plot. If there's x-axis, there's a y-axis. Very, very rare you actually have XY plots in biology. What you have are bar graphs, table bar graphs. Okay. And actually, this is it comes to one of the fundamental problems of difficult studying biology. Okay, that is because quite often, if you want to make XY plot, you need to know X. You need to know what is being changed, right? So X is something you're changing, and Y is the outcome. Okay, so often we say biology is not quantitative, but that's actually not the case. Anything you measure is quantitative. Okay, and that's why the Y-axis could be quantified. Right? And you have a bar graph. But often you do not know what x-axis is. Right? And actually knowing that x-axis is 80% of the problem. Okay? And as long as the data is in bar graph, there's really no model, no mathematics I know of can explain bar graphs. Okay? You have to have an x and then a y. Or any equation right now you have an x and a y. Right? So a lot of times, so biology, and then why it doesn't have x-axis? Because the nature of the experiments you do. A lot of time, you compare a healthy person and a patient. Well, the whole point is you do not know what's wrong with it, right? You measure, okay, so there's a two bar, one is a healthy, one is not healthy. How do you put on the x-axis? I mean, if you can put on the x-axis, you solve the problem. You know what's wrong with it, right? So this is quite often what you encounter in biology, and I would say, biology historically it came from. Historically, biology is zoology, right? So people look at birds, how big their eyes are, and how big their nose are in that's zoology, okay? And then when microscopy was developed, then people look into the cell. They say, well, here's a Golgi body, here's ER. That's still zoology. It's descriptive. You as you look into the microscope, you draw a picture, right? It's a descriptive. The biology is descriptive in nature. Okay, then and in the quantitative in terms of maybe putting down some bar graph, right? And nowadays, even with all the advancing technology, we can measure many things. So nowadays, like 19th century, since 1950, the difference between the past zoological approach is that now we know cells are made of genes, and protein are directed by gene expression, and then so what people can measure a lot about what response in terms of how much protein is making, how much modification is being made, and all that. But fundamentally, it is still zoological in essence, because what do you do with all that stuff? They're no different fundamentally from how big the eyes of the birds are, how big the nose are. You're just looking at more category, right? So anyway, that's my biased view of looking at the biology. But it is true. If I'm talking about at the operational level, okay, that's you're dealing with many categories of things, and you're describing it. That's a study of the mainstream study of biology, right? So now let's talk about interface of physics and biology. So far, I would say the first wave of interaction of physics and biology is really in the 1950s, where a wave of physicists from studying quantum mechanics going to biology. So Max Delbruck is the prominent example, which look at the phages, and he got a Nobel Prize by understanding the mechanism of the phage. Phage is a virus or bacteria, okay, using quantitative approaches. Okay, so that was the first wave of quantitative biology in the 1950s or 60s. Then in the 70s, the biology took off because of they discovered the power of molecular biology. You don't really need to understand things. So before it was really tough, right? And to think about what to do to do things. But then with the technology came through in the 70s, molecular biology, and they can just measure many things, right? And do brute force mutation and discover other things, right? Become a more discovery-based thing. And the physicist's role has mostly been in studying components. So physicists will study, membrane, the property of a membrane. Obviously, very important. Physicists will study transport, say, the channels, how ions go through. Physicists study coding of a protein, right? So, and of course, anybody who study whatever they're studying, they tell you how important it is. And everything is important, for a cell to work, for a biological organism to work. It's so amazing, so complicated, right? And you mess up something, it doesn't work, right? But take a step back. Relatively speaking, if you have the task of figuring out how a car works, right? Most of in the 1970s, most of what so-called biophysics would be like studying what the tires are made of. Okay, how the battery works, right? That sort of thing. Not touching really on the essence of how a car works, right? How the essence of how a car works is really, well, to me, the essence is the heat engine. That's the heart, right? How the cycle of a combustion leads to mechanical motion, eventually get this, right? And that's what makes a car. And the rest of you can, well, obviously you need tires to do, but that's the application, right? So I would say in the 1970s, a lot of the studies the physicists were looking at to look at periphery components. Okay, and that's biophysics. And then, but then technology keep on going, and biology has really experienced the power of technology. Because the more they can see, but the biology is infinitely complicated, right? The more you can see, the more you have a chance to discover something that's important for living. Okay, and so this trend continues through the application of technology to biology, including these days super resolution microscopy, right? And optics is a very important tool. And given the descriptive nature of biology, the more they can see, the more they can write down, right? And one of them possibly could be very important. So I would say that's the mainstream of a physics is the way physics interacting with the biology, right? It is at the component level, and I would say fundamentally the result that you obtain from this type of approach is it's not changing the descriptive sort of approach to biology, but providing more quantitative quantitative version of the description. So in this general area nowadays we call biological physics. Okay, and don't get me wrong, it's very important, right, for many things to have a good quantitative marker or something. Okay, remember, what enabled Kepler to develop his law was Kepler was standing on the shoulder of a giant, and the giant is a Tycho Brahe, who developed the instrument, the microscope, and meticulously recorded where the stars are in different positions. Right, and okay, so certainly it's good to have a good quantitative numbers or measurements on these things. Okay, but that itself, right, is still remaining at the descriptive level. Right. Now, I would say there is mainly in the last, starting in the after 2000, there's another way sort of physicists are interacting with biology. Okay, and that's what I call, it doesn't even have the word physics in it, it's called quantitative biology. Okay, and so what that is going after, right, it is to kind of understand biology more the way that the early pioneer in physics tried to understand the physical world. Okay, so that is to think about what Kepler did, what Newton did, right, was to look at simple systems and discover patterning them, right, and write down mathematical the description. Okay, and okay, so this often is it may be difficult to distinguish between writing down a model. They often say, oh, there's a phenomenon, we write down a model, right? You can write down a model that includes lots of detail and reproduce a phenomenon. That doesn't mean you understand it, right? So we believe, okay, everything eventually can come down to quantum mechanics, right? So if we have a big enough computer, we can in principle write down a Hamiltonian to describe every possible interaction, and you crank it, and let's say you produce the phenomena. Doesn't mean you understand it, other than that, okay, things run by quantum mechanics, right? So when we say understanding, in physics, when we say understanding, right, we mean a way of thinking about something, right? So that we can, so I often give two criteria for understanding something. If I understand something, then if you ask me a question, right, I can give you an approximate answer just with a few scribbles. And if you ask for a detailed answer, then I know a way to get to a detailed answer. You see what I'm saying? Right. So this is, I think, you ask me a phenomena without doing too much work. If I know how to think about it, I can pull out the answer. I have a direct way to think about it, so I can give you all the magnitude estimate and write down the answer. Right? And but if you want me, well, in detail exactly how this happened, well, then I know at least I can do calculations, perturbative calculation, this and that, to get to a more refined answer. Right? So that's kind of the two, and the biology is far from that. Okay, but still, could we find something that a simpler way to think about it to get to some sort of answer? Right, and that's sort of what quantitative biology is taking on. Not necessarily looking at all of the details, right, but have another way to look at a system and see whether the phenomena can be tied to some mathematics, right? Just like remember when Galileo was looking at things, when Kepler is looking at things, they didn't know lots of detail, but they're seeing some phenomena and they can associate the phenomena with mathematics. Okay, so there are people that believe biology cannot be described by mathematics. But now we're having more and more examples where biology can be described by mathematics. The phenomena, I do not mean model, I mean phenomena. Okay, because when you have a phenomena that is describable by mathematics, then there's a hope to understand it in a simple way. Okay, because you can have the same, everything, as I said, everything following from quantum mechanics. You can have just you do something, just random things come out, then this kind of thing is not worth studying. I mean, in terms of from a physics perspective, because it's just detail, right? You know, you need to know every component. Okay, but then if you think about it, if it's like that, how can biology handle it? I mean, a cell is not infinitely powerful. Okay, I mean, like I study bacterial cells. There's very limited things it can do. Okay, but then how can it survive to deal with so many different situations? Right. So then and that becomes sort of a question in of itself, right? Is there some secret that we don't know about just by looking at it, thinking about everything is just coming from molecular interaction? Of course, everything coming from molecular interaction, but is there something transcendent, right? So it's like in gas laws, you have a 10 to the 22 molecules colliding. Who would think that the PV equal to nRT will come out? Right, so that's sort of the approach a number of us are taking, and the quantitative biology program at UCSD is to cultivate this type of studies. So I've been we started this program 4 or 5 years ago, right? And then we are continuing. Well, this is a graduate program, unfortunately, we haven't extended to an undergraduate level yet, but maybe at some point we should. Okay, so it's already quite late, but maybe I show you guys have questions. We can talk about that first. Maybe I show you one piece of our actual research that you get a concrete feel of it. But do you have any sort of general questions we want to discuss first? Okay, so I show you a piece of our research. You see my PowerPoint?
Yeah, we do.
Okay, good. All right. So, so my lab, we study bacteria, okay, and I study two aspects. One is, so bacteria is the simplest kind of a living, free living organism, and it's the best characterized at component level. So we study bacteria so that we don't have to deal with missing components, and then the whole game is about how these components work together to make a living system. Okay, and so we have two aspects. One is how the molecular, so there are thousands of molecules inside a cell, right? And how do they work together to make a living entity that replicates and keep on going as long as there's food outside. And then another aspect is, so that's from molecule to cell. Another aspect we study is you put these cells together in some environment, and they will work together and to form a coherent community that's stable. Okay, so that's a two. So the latter part is called microbial ecology. The first part is cellular physiology, molecular biology. Okay,
Professor, your screen is, like on my screen, it's just a really narrow white line. Can you try resharing?
So the PowerPoint you see is narrow?
Yeah, just like a narrow vertical white line.
Can you?
Now it looks fine.
That's fine. Okay. So
I don't know what happened.
Okay, it's a screen problem. Right. Okay. Good. All right. So I tell you a piece of a story so we've been spending the last 5-10 years looking at the link from molecule to cell. Okay. And then more recently, the last 3-4 years, we started to look at how the cell works together to form a community. Right. But I share with you a piece of work on the first part. Where's the notes? Whoops. Huh? Yeah. I'm having a hard time. There it is. What do you see? Do you see the presenter mode? Or
Yeah, we see the presenter mode right now.
How's that?
Perfect.
Okay. All right. So I'm going to tell you a piece of work where in my lab, the students and postdocs are looking at the bacterial growth, and they discovered some laws, right? So bacteria, they're not just kind of blindly following quantum mechanics, but then they actually have their own organization, their own state. Okay, and in particular, you should pay attention. You should cheer when you see that they're actually XY plot. Okay, and that's the reason very simple. That's the hardest thing is to find the x-axis. So okay, very quickly say so we study E. coli, so one of the best characterized bacteria, right? And if you put a drop of E. coli cells into medium with some carbon and the nitrogen, then after a while they start growing exponentially. Okay, and eventually they run out of food and they stop growing and then reach a stationary phase. Okay, and during the exponential growth, then we have simple equation. The MDT is lambda, and where lambda is the exponential growth rate. Now this is not the XY-axis I'm talking about. This is okay, exponential growth. That's fine. Everybody know population can do that. Okay, but then it's interesting what this growth rate depends on. Like you feed these cells with different carbon sources. Some it grows rapidly, half an hour per doubling. Some it grows very slowly, 10 hours per doubling. So you can have orders of magnitude difference, okay, depending on what you feed it. And this growth rate depends on if you go to high temperature, low temperature. It clearly depends on temperature, in an environment, pH, osmolarity, many things. A higher drug, right? The whole thing about antibiotics is slow down the growth. Okay. So to understand exponentially growing bacteria, so what it means is to understand what determines how fast it grows. And so that's a concrete question, right? So I said the environment affects its growth, but then genetic factors, the enzyme that's expressed, regulation, all of these things affect how fast a bacteria grows, right? So you think about what goes into replication of the cell. Well, what is a cell? You broke up open a cell, then you see DNA, proteins, many things, right? And roughly speaking, 2/3 of the cell is water, 1/3 is biomass. Actually, this is remarkably conserved across biology. Okay, about it's 1/3 of a cell is a biomass. If you're looking to biomass, then what is it? Mostly it's protein. So the cell is to a first approximation of bag of protein. Okay, 2/3 of biomass is protein, and then there's RNA. Okay, and so basically it's the mode of protein RNA. And then most of these RNA is in the form of ribosomes. So ribosomes are machineries that make protein. Okay, so ultimately this thing has to replicate itself, right? So the whole thing about living system, it can under conditions to replicate itself. And what does it do since most of it is protein, replicate itself means replicating proteins. Okay, and replicating proteins is done by ribosomes. That's these special machineries. They're the size of 50, 100 proteins. And they come together to form the function of making the polypeptide chain that makes protein. Okay. Now, so I'm going to skip lots of details. These are the things you probably learn. If you haven't taken a biology class recently, you probably had it in high school biology. And the details are not important, but just that this process is extremely complicated. It involves many players. Ribosomes made of RNA and the proteins. Most of the RNA is in the form of actually ribosome, and then so we can skip all of these things. Okay, and the ribosome is a machinery that has a different RNA goes in and then stitched together the individual residues called the amino acid that stitch together protein. Okay, and you have 20 types of amino acid. So all of these amino acids need to find their respective transfer RNA to be fed into the ribosome ribose in order to make the peptide. Very complicated. Okay, details not important. All I want to show is that it's a very complicated process. Okay, and lots of details are known at the biochemical level. You can even write down rate equations for given the concentration. A is an amino acid, RS is the enzyme that stitch these things together. you can write down how fast this reaction goes. Okay, so somebody has studied all these things for you. Okay, and so if you think about it, then there are already hundreds of variables because the 20 amino acids each have its accompanying this and that. Okay, that's 100 variables and a couple hundred parameters. Because each one of these reactions requires a couple of parameters. Okay, so yes, you can write down equations to describe that, but most of the time we have no idea what the parameters are. Okay, even when parameters are measured, they are measured at 4 degrees Kelvin, 4 degrees Celsius, at whatever some weird condition that's convenient for measurement. It's not what you need for what's happening inside the cell. Okay, so that's a problem of parameters. The first taste of problem of parameters. Then, well, how do you get this amino acid? If you feed the cell with glucose, then there's complicated reactions that takes glucose to one of these amino acids. Each of the 20 amino acids has a particular way to be made. So that's biochemistry. So those of you who had having biology classes, it's a very boring biochemistry. Right? A lot of detail to study. Very important, right? Sometimes diseases due to something goes down. Okay, but lots of details. Okay, so there are 20 such things, and when you add up those, there are many hundreds of enzymes, many hundreds more metabolites, many thousands more parameters. Right, and that's not all, because the regulation is very important, because the amazing thing about a living system is that they can adapt to conditions. You're not always going to have a constant condition, right? It's the same temperature, food source. Food's going to run out, food gets switched, temperature changes, these guys excrete acetate and the pH buildup, right? So many things are changing, and they have to regulate these programs. So regulation, when you encounter regulation, many more parameters. Okay, and all of these things depends on physical environment, right? Temperature. You change temperature, every single one of these rates change, right? And if you want to know disease, you have one mutation, this rate change. Okay, so when you put it together, you're talking about many parameters, just to study the simple thing about bacterial growth, right? So this saying the summary of that is that you need millions of parameters just to describe exponential growth of bacteria. Now, imagine you're studying something more relevant, right? Now you're talking about modes of parameters, right? So it's not that, well, in principle, we believe the biochemistry works under this kind of a level, and then so if you know all the parameters, yes, you can make a model to make all this, but you can't because you don't know these parameters, right? So now let me ask if those of you who had a couple of years of physics, can you think of a case in physics where you have a problem with modes of parameters that you have no hope of characterizing, yet you can write down the result without knowing about the modes of parameters? Think about in physics. There is hope, right? And that's what we need to look up to. Let's see, junior, anyone beyond junior? Most of you, how many people have taken thermal?
We took 4B.
4B, okay, well, 4B doesn't quite get to that, right? So in what physical system we have this situation? Well, the situation is a gas. If you try to describe the motion, the property of a gas in the room, right? There are 10 to the 23 molecules, okay? And if you want to predict how this system of gas behave, if you start with Newton's mechanics, right? 10 to the 23 parameters, 23 molecules, you know Newton's law, each degree of freedom, you need two initial conditions. The initial position, initial velocity. Okay, that's 10 to the 23 initial condition. Okay, but that's nonsense, right? Obviously, you know, you can know about the property of gas without knowing exactly at some time zero of where this every particle is and what the velocity, right? So then, and then so of course there's alternative approach in thermodynamics, right? Very simple, PV equal to nRT. It describes the system without the need to know about the 10 to the 20 parameters. You only need one parameter, but R, the gas constant. Okay, so we're gonna think about, well, how does that come about? Right. So this is called dimensional reduction. You have it's apparently a very complicated system. Okay, but the behavior is captured by a few variables. So people try to derive Newtonian mechanics. Ultimately just come from gas collision. There's nothing more than that. You can make these days you can write a simulation program. You put 1,000 particles, they collide, and you get a gas, right? But this is the but then so Boltzmann tried to do this. He failed. Boltzmann, the great giant of 1800 physicist, right? He failed, and he killed himself. He committed suicide because he knows this structural mechanics that have this, but he couldn't get it from Newtonian mechanics. Okay, so it's not given that, right? Even for a simple system like that, it's not that we know about details. You can just predict what's happening in the larger scale. Okay, in physics, what we do is we study the large scale on its own, right? So historically, Newtonian mechanics is studied by itself. Thermodynamics is studied by itself. Okay, and then what happened was then Boltzmann that come together, the statistical mechanics, right? So statistical mechanics is a shortcut that takes what you know at the molecular level and bypass running all of the 10 to the 23 equation and immediately get to the thermodynamics. That's one example we have in physics, right? Where we can shortcut all the complications and go to the macroscopic behavior. Okay. And okay, so this is a bit more mathematical for those of you who have the statistical mechanics. The key there is that even though in principle the property of a gas depends on all of the variables of 10 to the 7 particle, what it really depends on is only one quantity, is the energy of the system. And that's the basis of Boltzmann's statistical mechanics, is that it depends on all of the variables through a simple quantity, the energy. Right, so then the challenge is to go from molecular cellular biology, sort of like the Newtonian mechanics in this picture, to physiology, right? And so our lab, what we did was actually to make that connection in the context of bacterial cells. Okay, and and in particular, take this kind of a details on the upper left, right, and link it to very simple behavior, the exponential growth, short-circuiting all of the thousands, millions of parameters that's needed to get this. Okay, and so I'm probably how much we are supposed to end. Okay, so we'll have do another. I'll take 15 minutes to end, and I'll give you some chance to ask questions. Okay, so and the key is basically these days in biology, there's lots of study at the molecular level, but there's very little study at the higher level. So in biology, this is called physiology. Physiology is about the property of the whole. Okay, and if you remember in the development of thermodynamics, okay, the whole is thermodynamics, right? And that is about when you increase temperature, pressure increases, things like that. Okay, you do not know anything about molecules. You just study the property of the whole, and you see the patterns. The whole happens to have simple relations, right? And that gives you a clue of how to connect to the body, right? And another example of studying at the microscopic, that's called phenomenology, right? Another example that you must be familiar with is say Ohm's law. Okay, when Mr. Ohms was studying electrical circuit, he had no idea electron, quantum mechanics that really describe the electron. Okay, I mean even now, right? Deriving Ohm law is a non-trivial thing. Okay, but it doesn't matter. You apply the voltage across the resistor, you get a current. You see the proportional. You write down Ohm's law, and the entire material is collapsed onto a single parameter, this resistor, right? And knowing that, never mind where it comes from, you can start to put circuit together and do things. Okay, so that's the phenomenological approach, and that's the approach my lab is taking to study biology. Right, and so just give you an example. Right, and then we found something similar to statistical mechanics, where the very complicated biological molecule, very complicated things, it actually exerts the effect through something that's quite simple, and biology captures that to do its regulation. Okay, so I want to talk about laws in biology. So this is one of my heroes, Ole Moller, is a Danish physicist, sort of grew up at the same time as Niels Bohr when the quantum mechanics is happening. Okay, so this is a biologist, and so here's a plot where the many circles. This is all about growth of a bacteria, E. coli, Salmonella, the simple bacteria. And so each growth is an exponential growth. Each point is about exponential growth, where if they feed things with different material, feed these cells with different food. So the upper right is one type of food and feed that is growing very fast, 1.8 doubling per hour. Okay, and the lower left is they feed some medium that the bacteria grow very slowly. Okay, and back then they didn't know what to. Then there's a whole lot of things they can measure. So they grow up an exponential culture of bacteria. They take the bacteria and they just measure the total RNA and total protein in this soup. Okay, and they take the ratio of RNA to protein, and you see there's a straight line. It's amazing. Right? So I was very frustrated by I had already at that time studied 5 years of biology, just more and more detailed, more and more complicated, right? And then by chance, I ran across this result. What was so amazingly simple was there's got to be something behind it. Okay, so you think the thing about biology is that it doesn't impress anybody if you find something complicated. Biology is very complicated. But when you see something simple, well, then there's usually a reason behind it. You can ask. It cannot just be randomly you have something simple. Okay, so saw this thing that's very simple. Well, they saw this thing that's very simple. Okay, relation. What does it mean? Well, RNA is mostly ribosome, as I was describing, and the protein basically is proportional to cell volume. So RNA protein ratio basically is telling you about ribosome concentration. So what it's saying is the faster cell growing, the more ribosome there are. But not just a qualitative statement, it's actually there's a mathematical relation, right? Wow. Bacteria can draw a straight line. That's amazing, right? And so why is that? So Moller already had a pretty good understanding of where this comes from, right? He said, well, if most of the I told you that ribosomes is the machinery that makes protein. Okay, if all of this machinery are engaged in making proteins, okay, they're all working at maximum rate. Then if the cells want to grow faster, then you need to have more machinery to make this protein. That's all. Okay, and so mathematically, you're just saying the NDT, M being the total protein mass, right? We know empirically it's a lambda times M. That's exponential growth, right? But then all of the increase in protein is coming from the ribosome, that MRB, right? And multiplied by some rate they're working. Okay, so if ribosomes are making all the proteins, then you just equate these two, and you see the fraction of ribosome over total protein should be proportional to the growth rate. Okay, this is a very simple explanation for this. And you can check this and the quantitative and numbers sort of the slope correspond to the measure how fast ribosomes work. Now, okay, so they're saying most ribosomes engage in translation. This result, very striking result, sat there for about 50 years. Nothing much more happened to it. So when we came across this result, so Max Scott is the first author of this paper. It's one of my sort of most cited paper in our lab. And so we were wondering, so looking at it from a physicist's point of view, it seems like it is missing a companion relation. Okay, what I mean is that, so this is saying, okay, you have ribosome working at a certain rate, and then the faster, so the more nutrient you feed it, the faster it grows. Okay, but then so we say, okay, this is so we try to think about orthogonal perturbation. This is a perturbation where things are varying. When it's varying in the experiment, when it's varying the nutrient that's applied to the cell. Then we ask in the spirit of physics, well, what if we fix the nutrient which change how fast ribosome work? And this you can do by applying antibiotic. Okay, and when we do that, we're very surprised to find we got another straight line. Okay, that is so we inhibit we applied a little bit of antibiotic to slow down how the growth of the cell. Growth rate slow down. but then it makes more ribosome. Okay, that's the upward going line. Okay, details not important. We have two ways of varying the growth of these by limiting nutrient or by limiting drug, by applying drug, and the response is completely different. Both straight line, but different. So then we get curious, well, is this happening just to ribosome or to other stuff? Also, the other cellular protein. So we looked at a very well-known transporter that takes in food for E. coli. Well, and then we were so surprised, a straight line. That is, so during nutrient limitation, when ribosome is going down with decreasing growth rate, the transporters were increasing with a decreasing growth rate. And if we apply drug, so the ribosome increase, then the transporter decrease. We were so fascinated by this. These are very simple experiments, could have been done in 1950. Okay? But it's just drawing straight lines left and right. And biology is just not supposed to do this. Okay? Okay, so then we know we're dealing with some system with regularity. Right? There's no amount of people claim we know everything about E. coli. There's nothing that from the molecular wiring can predict this kind of a relation. All right. So then this led us to propose a very simple model. Okay. So I'll describe the model to you. So we say, okay, in a cell they have all kinds of proteins. So this is a coarse-grained model. We have all kinds of protein, and we call it as a pie. Okay. Among the pie, there's a portion called R, the green guys, that are ribosomes. They're responsible for making other proteins. Then there's a portion called C proteins. They're responsible for taking up things, right? And then there's a portion called A protein, therefore synthesis. Okay, and so to grow fast, the more ribosome you have, the faster you will grow. The more uptake protein you have, the faster you will grow. The more biosynthetic protein you have, the faster you will grow, in principle. Okay, but they're constrained because they have to add up to 100%. Well, it turns out half of the cell is allocated to housekeeping stuff. You can actually. Okay, so they have to add up to about 50%. This system completely explains this very simple equation, 4 equations, right? Linear equation, completely explains the observation and more can make prediction. Okay, so an analogy to this is a simple resistors in series. Okay, so if you imagine you have a circuit with 3 resistors, then and you think about say current. So in this analogy, current going through the resistor is the growth rate. That's the flux, neutron flux going through. Okay, and then you think about the voltage across each resistor as the phi. Phi is the fraction of the protein that's devoted to a certain function. Okay, then you see basically all we're getting is this Ohm's law. Okay, the relation between growth rate and how much protein there is just Ohm's law for each individual element. As the more protein, to push more flux, you need more protein. Okay, but the overall amount of protein is constrained, so you have this relation. Okay, so it is the detail doesn't matter, but there's an attempt now to try to put together the behavior into a very simple model. Okay, and then we were able to I'll skip these. So then we're able to test these things by forcing the cell to make useless proteins, and basically things just agree with what this model will predict. So this is one case we can make prediction of biological system with 0 parameter. We don't need any parameter to do this kind of prediction, right? So then this leads to the strategy of control, which I would like to just spend the last few minutes talking about. Okay, so the basic picture is we have nutrient coming in. Okay, they are transported by these uptake proteins, C proteins, and turned into a pool of central metabolites, amino acid, ATP, and all that. Okay, then the amino acid are stitched together by ribosome to make proteins. That's a basic kind of a year worth of a cellular metabolism boiled down to a simple picture, just that. Okay, all these proteins are converting external food, glucose, xylose, whatever, into amino acid, and amino acids stitched together to make proteins. Okay, and how much that flux is depends on the concentration of each type of protein in the protein. Okay, and in there is this key variable that is for a given amount of ribosome, how fast the ribosome works to stitch the protein together. So there's a translation rate called sigma. Okay, and then gene regulation determines how much of the ribosome output goes to the C protein, and then the R protein, the ribosome, and so forth. Okay, now the key here is how much ribosome makes ribosome. That's what determines a growth rate. That's a linear relation I showed you from the beginning, right? More ribosome, faster growth. So if you look at molecular biology, they tell you, oh, ribosome control is very delicate, as controlled by this magic molecule called PPGPP. By the way, this is for bacteria. Similarly, very complex control of a ribosome is in mammalian cells, in, say, in particular having to do with cancer. So people that study cancer, obviously proliferation, controlling proliferation is very important, and ultimately it all boils down to ribosome making more protein, right? So this kind of a study is for any organism is a key thing. How does ribosome decide how much ribosome to make, how much other things to make? Okay, and this in these bacteria, it's oh, there's a magic molecule. Never mind what it is, a weird molecule that controls how much ribosome works. Okay, and then there's a description on which protein sort of makes it senses these pool of metabolized tRNAs and so forth. And depending on the idea is if the metabolites are in shortage, right? So if the nutrient coming in is small, then the metabolites, the ATPs and AA will be low, and then you want to dial down ribosome synthesis. Okay, and when the metabolite level is high, nutrient high, you want to make more ribosome. That's kind of the idea of the control. But there is a problem, is that how does it know about, because there, as I said, hundreds of molecules, thousands of molecules determines eventually how the ribosome works. Okay, and you limit any one of them, it wouldn't work because it needs all of these things to happen together. Okay, so they have hundreds of parameters, thousands of parameters that eventually go into this decision making. But how is it possible for the cell to deal with thousands of these parameters? Okay, and that's why I remind you in the beginning of showing all of this stuff is happening, right? And how would a cell know when something is missing? Where is it missing? So then, so in another word, is the cell, in order for it to respond to the environment, it needs to know how fast it can grow, how fast it is growing. But if you think about a cell as a bag of molecules, how does a bag of molecules know how fast it's growing? Okay, so there are the number of fundamental questions like that. Let's transcend detail. How does a bag of molecule know how big it is? How does a bag of molecule keep time, right? How does a bag of molecule know how fast it's growing? Because all of these properties is an emergent property of the whole. It's not just molecules, right? So there's a fundamental question here, right? And then we had a guess that actually the cell is not monitoring actually every one of these molecules, but it's monitoring how fast the translation process itself is proceeding. That is, it's monitoring the translation rate. Okay, so instead of all of these bag of molecule MI directly affecting the control, it is done actually through translation rate, which itself depends on all of them. Okay, so it's detecting the process rather than looking at individual components. So that was our guess. Okay, and the guess turned out to be right. I'm going to skip some. So this is the analogy to Boltzmann mechanics. That is a system that seemingly depends on many other things. It actually depends on many things through one thing. Okay, and that kind of strategy is basically a guarantee for dimensional reduction. And so we can write down some equation. We end up with a single ordinary differential equation that can predict the behavior of the cell. When you switch medium, switch nutrient, and then you can go to speed up in growth, slow down in growth, right? It's almost like a Newtonian mechanics type of equation that describe the growth of a cell, right? And then this is just the example of how it works, right? So the three different colors are basically the different quantities. Green is the ribosome, red is the uptake proteins, and black is the overall the cell numbers, cell mass. And you can see that. So this is a prediction is the line, and the data points are the experimental result. No fitting parameter, the entire description, right? By solving a single equation. Same equation. If you reverse so this is a nutrient upshift. That is, it start initially growing a poor nutrient conditional. Is the experimental situation. Now you give it a boost of a nutrient. that is strict to fast growing. Exact same system if we just reverse, start with the fast growing nutrient and then run out to a poor nutrient. Same equation, just switch the initial and final state, and you lose to this kind of prediction. And again, just dead on, right, in terms of it. So we have here then at least one case where we can really predict the dynamics of the system, right, without knowing the detail, just by looking at, starting from this macroscopic levels, right? And there's a measurement about internal state of the cell. I'll skip. Okay. So finally, let's see. Yeah. So so this is about we assume that this control is given by translation rate. So they actually recently student actually did the direct measurement and showed experimentally that the signal is indeed proportional to the translation rate. So the final carry-home message is that these cells are very clever. They invented a mechanism to tell how fast the cell is growing by directly detecting the activity. So to give you a sort of a example to carry home, is that suppose you're running a big operation, a big factory, right? That requires many inventory, many sort of buying many components, and you're putting together and you're selling things. Okay, how do you make this factory operate in an optimal way? That is, you want to hire just enough workers so that you can depend on the demand, of course, right? If it's a good season, let's say we're making cars, many cars are being sold, then you want to have lots of input and hire lots of workers to do this, right? And if there's no order is low, you don't want to do this. Okay, now, but then it's not just a matter of the order, but it also depends on input. Let's say it's difficult to get hired. Well, then you obviously, then that's a bottleneck. You just cannot make that many cars, right? And so it's complicated decision making that depends on many components. So how do you do this decision making? So this is kind of the situation the cell is faced with, right, in making decisions. So ribosomes are the workers, right? How many workers should it hire to do this? Okay, so the traditional base is by looking at the two things called the product inhibition or the supply driven thing. So if one is a supply driven, is that if I have lots of input, I hire lots of workers to make stuff. Right. The other is that if I have lots of inventory building up, I cannot sell my car, then I stop hiring. Okay, so that's simple. That kind of a strategy works if you're talking about just one input or one output. There's a very visible thing that's output, a very just one thing that's dominating your input. But in a case where you have many inputs coming in, and you're selling many things, it's really not clear about how to weigh the different components. Okay, so in this example, then what's E. coli doing? Its decision-making strategy is very simple. It just look at the number of workers that's taking break. Okay, and the decision is simply, if we have lots of idling workers, it means okay, somewhere it's stuck. You can fire some workers. It's okay, right? And if everybody's working at maximum, they have no time to sleep. You hire more workers. Okay, use it or lose it. So that's what it's a very simple strategy bacteria use, and the end result is a very simple controlled sort of simple relations. Okay, so this is the example to show that in some cases we're seeing more and more of these cases that physics-like simple description of a complex system is possible, and biology is not just randomly made of a random collision of all the molecules, but it actually follows a very simple rules. The rules that we cannot imagine, but they know, and they can manage themselves very well. Okay. So yeah, of course, I leave the tough one to you guys. Right, so we study bacteria, and there's also eventually ones you can go on to study more complicated systems. All right, yeah. So this is a sort of an ongoing work from many students and postdocs. As I said, over the past 10 years, we've been studying this sort of systems. Okay, yeah. This is all I have in terms of giving you a flavor of what we do.